At the center of the brain, cradled between the two great hemispheres in a warm stillness of surrounding tissue, there rests a structure so small that the whole of it could be hidden behind a single grain of rice. It is unpaired, solitary, suspended along the midline like something placed there with deliberate care. It sits dorsal to the third ventricle, tucked into the epithalamus, held in the geometric heart of the skull at a depth of roughly 3 inches from any surface. The brain is a dense and complicated country, and this small thing occupies its exact center. It is called the pineal gland, and it has been there in one form or another for longer than the kind of skull that now encloses it has existed. To hold it in the mind is to hold something ancient. It is roughly 5 to 8 millimeters in its longest dimension. Its color in life is a soft reddish gray, faintly pink from the capillaries, threaded densely through it, fed richly by blood. It has no internal chambers, no obvious architecture visible to the eye. It simply rests there, quiet and warm and dark, receiving almost nothing from its surroundings except by a long and winding road, the signal that night has come. What it does with that signal is what everything else follows from. The word pineal comes from the Latin for pine cone, and the resemblance, slight as it is, was enough for the name to hold across centuries. The philosopher René Descartes, writing in the 17th century, believed it to be the seat of the soul, the one unpaired structure in the brain, he reasoned, must be the meeting point of mind and matter, the single locus of unified experience. He was wrong about that, but something in his intuition about its singularity was not entirely misplaced. Of all the endocrine structures in the brain, the pineal stands alone. It issues one principal signal into the bloodstream each night, and that signal, as it turns out, is carrying the most important message the brain has for the body, that the sun has gone down, that darkness has arrived, and that the long work of the night can now begin. To understand the pineal gland fully, it is worth traveling backward into deep time, because the structure sitting at the center of a human skull is not the same structure that sat beneath the skulls of the first creatures who carried it. The pineal has a history so long that most of what it once was has been quietly set aside by evolution, replaced not by something better, but by something different, a more indirect arrangement suited to the kind of brain that grew up around it. 400 million years ago, in the shallow warm seas and muddy riverbanks of the Devonian period, the earliest vertebrates were navigating a world defined by light. Not light in the way a human experiences it, processed through a complex visual cortex and layered with meaning, but light as a raw environmental signal, a constant and indispensable measure of the world's rhythm. Day was warm and visible, night was dark and still. The body needed to know which was which, and it needed to know continuously. The ancestral pineal was built for exactly that. In the lampreys, jawless fish whose lineage extends back close to that Devonian world, the pineal body is positioned near the dorsal surface of the brain, just beneath the translucent tissue of the skull. It contains photoreceptive cells bearing visual pigments closely related to the opsins of the lateral eyes. Light passing through the skin and bone overhead reaches those cells directly. The lamprey's pineal is in this sense a third eye, one that measures not form or color or motion, but the ambient luminance of the world above, the slow arc of daylight and the slow fall of darkness, tracked across the hours by a structure sitting in the center of the head. The same arrangement, with variations, appears in the frogs, in tuataras, in many lizards. In the tuatara, a reptile whose lineage has changed so little across 200 million years that its living representatives are genuinely archaic, there is a parietal eye set directly into the top of the skull, a small translucent opening with a lens, a retina, and a direct neural connection to the brain beneath. It perceives light and darkness, calibrating the animal's circadian timing by measuring the sky. It does not form images. It does not see in the ordinary sense. It simply watches the quality of the light and reports what it finds. For most lizards, the parietal eye is less anatomically distinct, but the connection remains. A small patch of translucent scale above the dorsal surface, called the parietal foramen, admits light to the pineal or peripineal structure below. On a flat sun-warmed rock in early morning, a lizard orients its body to the angle of the light, and somewhere beneath the top of its skull, the ancient photoreceptive tissue is doing its slow work, measuring the angle and intensity of the sun, tracking the season through the length of the day, setting the body's deep rhythms in accordance with the world outside. There is something very still about that image. A small animal on a warm stone, the light coming down through a patch of its own skull, the body reading the sky from the inside. It has been doing this in some form for hundreds of millions of years. In mammals, this arrangement changed, not all at once and not by intention, but slowly across the long expansion of the Mesozoic and the Cenozoic eras, as the mammalian skull thickened, as the neocortex grew to fill the space inside it, as the brain became large and metabolically expensive and increasingly layered over its old structures with new ones. The direct photoreception that served the lamprey and the lizard so well became impossible. The skull was too thick. The brain had grown too dense between the pineal and the surface. Light could no longer reach the gland directly. Evolution did not abandon the pineal, it rerouted it. In mammals, and in humans most completely, the pineal still receives its signal, but that signal now travels by a route of extraordinary indirection. It begins in the retina at the back of the eye, where specialized cells detect the ambient light of the environment. It travels through the optic nerve, and then to a small structure in the hypothalamus that serves as the master clock. From there, it descends through the brainstem, passes into the upper thoracic spinal cord, and then climbs back up through the neck by way of the sympathetic nervous system, arriving at the pineal by the root of the carotid nerve plexus. Slender nerve fibers traveling upward along the great vessels of the neck, threading their way into the skull to reach the gland at its center. The signal that arrives is not light itself, it is a neurotransmitter. The pineal no longer knows the difference between a clear afternoon and a rainy one, does not sense the color of the sky or the angle of the sun. It knows only one thing: whether the nerve fibers arriving from the neck are active or quiet. When they are quiet, when the sympathetic signal is low, the gland rests. When they fire, which happens in healthy physiology in the hours after darkness falls, the pineal gland wakes to its particular work. This transformation from a direct light-sensing organ to an indirect relay receiver took hundreds of millions of years to accomplish. It is one of the great quiet reconfigurations in the history of vertebrate biology. The function was preserved, the mechanism was entirely rebuilt, and the pineal, which once looked directly at the sun through a hole in the skull, now waits in perfect darkness at the center of the brain, listening for the signal that tells it the world outside has gone dark. That darkness at the center of the skull is genuine. The pineal sits behind the blood-brain barrier, though, importantly, its secretory capillaries are fenestrated and open, giving it unusual access to the circulation, and it receives no light of any kind by any path. It is one of the darkest places in the body, surrounded on all sides by tissue and fluid and bone. The world the pineal inhabits has no quality of light whatsoever. It has only the message that light, or the absence of it, has produced somewhere else, transmitted here by a chain of chemical and electrical signals spanning the full length of the body. The cells that do the pineal's work are called pinealocytes. They make up the great majority of the gland's cellular content, somewhere between 80 and 90% of all the cells present. The rest are supporting cells and neurons, but the pinealocytes are what the gland is for. They are neuroendocrine secretory cells, a hybrid type that carries qualities of both neurons and endocrine gland cells, and they are built at every level of their anatomy for the task of releasing a signal into the blood. A pinealocyte viewed under a microscope is a cell of unusual shape. Its body is compact and relatively undistinguished at the center, but from that body extend long, fine cytoplasmic processes, slender projections that reach out through the tissue like threads, making their way toward the nearest capillary. The pineal gland is richly vascularized, receiving a blood flow that is among the highest per unit of tissue volume of any organ in the body. The capillaries are close, and they are fenestrated. They have small windows in their walls, openings that allow molecules to pass freely between the bloodstream and the tissue on either side. The pinealocyte reaches toward these capillaries with its fine extensions. The tips of those processes are positioned precisely at the capillary walls, where the chemistry, when the signal comes, will be released directly into the bloodstream. There is no distance to cross, no elaborate mechanism of transport. The molecule is made, the signal arrives, and the processes that have been waiting quietly all through the daylight hours release their contents into the blood at once. The pineal also communicates through the cerebrospinal fluid, the slow-moving clear liquid that bathes the brain and spinal cord, circulating in a gentle continuous flow around and through the central nervous system. Pinealocyte processes reach not only toward capillaries, but toward the ventricles, the fluid-filled cavities inside the brain, and the molecule the gland produces can be detected in cerebrospinal fluid at concentrations that follow the same nocturnal pattern as the blood. There is something fitting in this. The brain, floating in its fluid, bathes in the signal of its own nighttime chemistry, surrounded by the same hormone that tells every other organ that darkness has arrived. What the pinealocytes produce and release is melatonin, and the story of how they produce it, and when and why, begins with a quieter chemistry operating through the long hours of the day, a slow preparation that makes the night's signal possible. But before that story can begin, it is worth staying with the gland itself a little longer, because the pineal gland, over the course of a human life, changes, slowly, silently, in a way that leaves no symptoms and causes no pain. The soft tissue of the gland undergoes a transformation that has been observed in human specimens for centuries and is now known to be nearly universal. Calcium begins to accumulate. The process begins early, sometimes in the first decade of life, more commonly in the second. Small deposits of calcium and phosphate, crystallized into the mineral form called hydroxyapatite, begin to form within the pineal tissue. They are visible on imaging studies that detect calcium, on ordinary skull X-rays, and on computed tomography scans. They appear as bright points, angular and distinct, embedded in the soft gray mass of the gland. These deposits are called corpora arenacea. The name means sandy bodies, and under the microscope, they do have something of that quality, gritty, granular, made of a substance that belongs more to the mineral world than to the cellular one. They are sometimes called brain sand. By the age of 17, roughly 40% of individuals show detectable calcification on imaging. By middle age, the proportion is considerably higher. By old age, the calcification of the pineal is common enough to be considered a normal anatomical feature, an expected finding, unremarkable on the radiologist report. No one is entirely certain why the pineal calcifies. The best current picture is that the hydroxyapatite crystals form within the extracellular matrix of the gland, deposited gradually over time by a process similar in some ways to the calcification that occurs in other soft tissues under certain conditions. The pineal is exceptionally high blood flow, and its unusual extracellular chemistry may contribute. The calcification does not appear to require any pathological stimulus. It simply happens in most people across the decades. What the calcification means for pineal function is a question with a more complicated answer. There is some evidence that the degree of calcification correlates with reduced melatonin output in older individuals, though the relationship is not a simple one, and reduced melatonin in aging is likely driven by multiple factors, including changes in the sympathetic nerve supply, changes in the activity of the enzymes responsible for synthesis, and possible changes in the master clock itself. The crystals accumulating in the tissue are one part of a larger picture of slow change. But there is something quietly striking about the image of those crystals forming. They are among the most slowly accumulated things the body makes. The pineal gland that rested at the center of the brain at age 5 had none of them, or very few. The same gland at 40 has accumulated decades of slow mineral deposition, a layer by invisible layer, the crystals settling quietly in the soft tissue like sediment at the bottom of a still pool. The pool does not stop working. The sediment does not prevent the water from moving. The pineal, calcified and gritty and marked by the years, still issues its nocturnal signal on most nights in most people for most of a lifetime. The crystals simply accumulate around it, year after year, as the rest of the gland continues its quiet work. There is a version of this that appears in many other places in biology. The slow mineralization of living tissue, the way calcium finds its way into structures that began as purely cellular, the gradual stiffening of what was once entirely soft. Bone is the most familiar example, the great hard architecture of the vertebrate body, built up over years from the soft cartilaginous template of the embryo. The pineal's calcification is different from bone formation, but it shares the same unhurried quality. The body makes crystals slowly. It has time. The pineal has been accumulating those crystals in the human lineage for as long as there have been anatomists to find them. Ancient specimens show the same corpora arnosa that modern imaging detects. The gland calcifies, apparently, because it always has in every human being across recorded history. This is not a product of modern life, or modern diet, or any recent change in the environment. It is simply what the pineal does, in the long, slow background of its biology, while it goes on doing everything else. To rest attention on the pineal gland is to hold something that operates entirely out of sight and almost entirely out of awareness. No sensation corresponds to its activity. No feeling marks the moment it begins its nocturnal release. The body does not know in any conscious sense what the pineal is doing on any given night. It simply finds, some hours after darkness, that sleep has become possible. The heaviness arrives, the eyelids descend, the world recedes, and somewhere at the center of the brain, in a space no larger than a small seed, the gland that has been preparing all day long is releasing its signal into the passing blood, which carries it outward to every tissue and organ and cell, each one reading the same quiet message in the same molecular language. The signal is melatonin. The gland is the pineal. The darkness that prompted the signal is outside in the world. But the signal itself travels in the interior dark of the bloodstream, in a body already mostly asleep, in a brain already softening toward unconsciousness, quiet and warm, and receiving the chemistry of the night. The capillaries thread through the gland's tissue without pause. The blood moves through them at the same steady rate it moves through the rest of the body. The pinealocyte processes touch the capillary walls like fingers on a pulse, and what they release into that moving stream will be in the heart within seconds, in the extremities within minutes, in the cerebrospinal fluid bathing the brain before the hour turns. There is no drama in this. There is no visible event, no sound, no heat, no light. The whole operation proceeds in a stillness that is only broken 7 or 8 hours later, when the morning signal arrives by the same long neural path, and the chemistry reverses, and the night's work is done. The pineal rests at the center of all of it, small, old, darkly housed, and exactly where it needs to be. Its history stretches back to a time when the ocean was the world, and the vertebrate skull was a thin plate, and the ancestral photoreceptive tissue looked up at a sun that was already 4 billion years old. Since then, over the slow hundreds of millions of years, the skull thickened, and the brain filled it, and the direct path to the light was closed off, and the gland retreated into its permanent interior darkness. The function remained, the architecture changed, and what was once a window became something more like an ear, an organ that listens for the report of the light it can no longer see and acts on what it hears. The lizard on the sun-warmed rock still carries the older arrangement, the translucent window in the top of its skull, the parietal foramen admitting the light of the sky directly to the tissue below. On a morning when the rock is still cool from the night before, and the first light of the sun is flat and golden across the surface, the lizard turns slowly to face it. Something beneath the skull is measuring. Something old and photosensitive is confirming, from the quality of the light arriving through the bone, that day has begun. In the human brain, 3 inches in from any surface, in a space where no light of any kind has ever penetrated and never will, the pineal is receiving the same information, the same yes, day has begun, by a route of extraordinary indirection, by a chain of chemical signals that began at the retina and traveled through the hypothalamus and descended into the spinal cord and climbed back up through the neck and arrived here in the dark where the gland was waiting. The architecture of that relay, the extraordinary, winding, multisynaptic path from eye to pineal, took hundreds of millions of years to assemble. It replaced something simpler with something far more complicated in order to preserve a function that life apparently could not do without. The measurement of day and night, the internal record of the light-dark cycle, the chemical clock that tells the body where it is in the 24 hours of the Earth's rotation. The pineal holds that record and issues that signal each night, in the small warm dark at the center of the skull, in a space where time is measured in the chemistry of a single molecule, and the seasons are registered in the duration of its release, and the years are written in crystals of calcium slowly accruing in the soft tissue, bright and angular and utterly unhurried. Those crystals have been accumulating since childhood. In most people alive right now, the pineal gland at the center of their brain carries some small quantity of them, pressed into the tissue, embedded in the extracellular matrix, forming layer by invisible layer across the decades. They are made of calcium and phosphate, the same minerals that built the bones. They are among the slowest things the body makes, and among the most durable. Long after the cells around them have been renewed and replaced, the crystals remain. They do not interrupt the gland's work. The nocturnal signal still rises each evening, still moves through the blood, still finds its way to every tissue that has a receptor waiting for it. The crystals settle quietly in the background while the chemistry runs its nightly course, as it has run every night of a human life, from the first weeks after birth until very near the end. What accumulates in the pineal over a lifetime is not just calcium, it is time. Each year adds its quiet layer to what was there before, the way a still pool adds sediment grain by grain in the cold, slow dark of its deepest water, indifferent to the seasons above, keeping its own unhurried record of the years. The gland does not know this. It has no awareness of the crystals forming in its tissue, no record of the seasons it has measured and reported, no memory of the thousands of nocturnal signals it has issued into the blood of a sleeping body. It simply continues. The sympathetic fibers arrive each evening carrying the news of darkness. The chemistry responds. The molecule is made and released. The blood carries it away. And in the soft tissue between the crystals, the pinealocytes extend their fine thread-like processes toward the capillary walls, as they have done every night of a lifetime, as they will do again tonight. In the deep, still center of the brain, in the dark where the body's oldest light-reading organ now does its work entirely without light, surrounded by the slow mineral record of all the years it has already kept. Somewhere in the late evening, in the quiet interior of the brain, a threshold is being crossed, not with any sound, not with any visible change, only with the slow, measurable rise of a single molecule beginning to appear in the blood, first at trace concentration, then in a gentle climbing curve that will not reach its peak for several hours yet. The moment this rise becomes sustained, becomes committed, has a name in the science of sleep. It is called dim light melatonin onset, and it marks the precise chemical beginning of the biological night. The molecule crossing that threshold is small. It weighs almost nothing on any scale that would register in ordinary life. A picogram, 1/1,000,000th of a gram, dissolved in a milliliter of blood is enough to be meaningful. 100 such picograms gathered at the signal's peak are enough to reach every tissue in the body and tell them in the only language chemistry knows that darkness has come and held. The entire hormonal announcement of night is carried in concentrations so fine they would vanish entirely against the weight of a grain of salt. And yet the body reads them with absolute precision, tissue by tissue, organ by organ, adjusting to the message the molecule carries. Before that molecule can exist, something must be made, and before that thing is made, something else must wait. Deep inside the pineal gland, through all the bright hours of the day, a chemical sits in high concentration and does nothing in particular. It is called serotonin, and the pineal holds it in quantities that exceed most other regions of the brain by several times over, a reservoir held in readiness, though the day gives no instruction to use it. Serotonin arrives in the pineal along a biochemical chain that begins much earlier with something simpler still. From food, from the proteins in a meal eaten hours or days before, comes an amino acid the body cannot synthesize on its own. It must be obtained from outside. Its name is L-tryptophan, and it circulates in the blood until it crosses a specialized barrier and enters the brain. L-tryptophan is not melatonin. It is not even close at the moment it arrives. It is the raw material at the far beginning of a chain of transformations, each one carried out by a specific enzyme, each one producing a compound slightly different from the one before it. The way a stone worked through successive hands becomes, finally, something it never resembled at the start. The first transformation adds a hydroxyl group to the tryptophan molecule. The enzyme responsible is called tryptophan hydroxylase, and what it produces is a compound called 5-hydroxytryptophan. The name matters less than the image, a molecule slightly altered, one chemical step closer to something the pineal can use. That intermediate does not persist long. A second enzyme finds it almost immediately and removes a small molecular fragment, a carboxyl group, and what remains, lighter now, rearranged by that single subtraction, is serotonin. The same compound found in other regions of the brain, carrying other signals, performing other functions. Here in the pineal, it carries none of those functions during the day. It simply accumulates. It pools in the tissue, held at concentration, waiting through the full arc of daylight for a signal that will not arrive until the light has gone. That pooling is important. The pineal gland does not make serotonin slowly, moment by moment, precisely when it needs it. It builds a reservoir. The serotonin that will eventually become tonight's melatonin may have arrived hours before sunset, synthesized and stored while the sun was still high and the gland was receiving no nighttime signals whatsoever. In that sense, the day is already preparing for the night, quietly and without intention, the way a reservoir fills long before anyone opens its gates. The gates, when they do open because a signal arrives from outside the pineal. That signal will be addressed in its own time. What matters here is what happens inside the gland the moment the signal comes. Something changes, not the serotonin itself, not yet. What changes is the activity of an enzyme, a protein sitting in the pinealocyte, present during the day in low amounts, not yet doing much. When the signal arrives, this enzyme's activity surges. In some species, particularly nocturnal mammals, the surge is extraordinary, activity rising to 50, even 100 times its daytime level within the span of a few hours. In humans, the rise is more moderate, but it is still unmistakable, still precisely timed, still the pivotal event on which everything downstream depends. This enzyme is called arylalkylamine N-acetyl transferase. The name is long and the function is specific. It takes serotonin and attaches an acetyl group to it, converting it into a new compound, an intermediate one step removed from the finished molecule. The enzyme is the rate-limiting step of the whole synthesis, the narrowest point in the chain, the place where the pace of the entire process is controlled. When this enzyme is active, melatonin production rises. When it is inactive or degraded, melatonin production stops. Everything before it in the chain can proceed, and the serotonin pool can be full to capacity, but without this enzyme working, the night's chemistry cannot advance. The intermediate compound it produces, N-acetylserotonin, is not melatonin either. It is one step short, but now only one step remains. A second enzyme, already present in the pinealocyte, finds the N-acetylserotonin and performs a final modification. It is called hydroxyindole-O-methyltransferase, and what it does is transfer a methyl group, a small chemical fragment donated by a compound called S-adenosylmethionine, onto the N-acetylserotonin molecule, attaching it at a precise position. This single addition, this small methylation, completes the transformation. What emerges from that reaction is melatonin, N-acetyl-5-methoxytryptamine, the molecule of the night, fully assembled inside the pinealocyte that made it. 2 enzymes, 2 steps, one narrow gate and one finishing touch, performed in sequence inside a single specialized cell, in a gland the size of a cherry pit, in the center of the brain. The entire hormonal announcement of darkness is the product of chemistry this compact, this precise, this quietly executed in the dark. Once made, melatonin does not stay in the cell that produced it. It passes out through the pinealocyte's membrane with relative ease. The molecule is lipid soluble, meaning it moves through fatty membranes without difficulty, and it enters the bloodstream through the fenestrated capillaries that run close by. The gland is richly supplied with blood for its size, and the melatonin it releases is carried away almost immediately, dispersed into the circulation, beginning the wider journey that will bring it in time to every organ and tissue that carries receptors suited to receive it. Some of the melatonin released by the pineal does not go into the blood at all. It enters the cerebrospinal fluid, the clear liquid that bathes the brain and spinal cord, through drainage into the nearby third ventricle. The concentrations of melatonin in cerebrospinal fluid are higher than those in the bloodstream, sometimes considerably so. The brain, lying close to the source, receives a more concentrated signal than the rest of the body does. In the quiet fluid that surrounds the sleeping mind, melatonin is present at higher levels than anywhere the bloodstream carries it. A chemistry of night concentrated at the center of the structure, it is most directly concerned with timing. The rate-limiting enzyme, the one that controls all of this, has one more property worth dwelling on. It is not a stable molecule. When it is active, when the night signal is sustaining it, protecting it from breakdown, it performs its function reliably across the hours of darkness, and the melatonin output it drives continues steadily, reaching its peak sometime between 2 and 4 in the morning in most adults sleeping on a conventional schedule. But the enzyme's stability is conditional. It depends on continuous signaling. If that signal is interrupted by light, by a disruption in the pathway that carries the nighttime message to the pineal, the enzyme does not simply slow down, it dissolves. It is broken apart by proteolytic machinery inside the pinealocyte, degraded within minutes, reduced to fragments that carry no enzymatic activity. That degradation is not a flaw, it is a feature. The system is built to be interruptible, reversible, responsive. It is not enough to make melatonin in the dark and then hold on to that chemistry passively through whatever comes after. The molecule of night must be produced continuously under active authorization for as long as the night lasts, and it must stop sharply, quickly, the moment morning comes and the light signal arrives to withdraw its permission. A stable enzyme, one that persisted regardless of ongoing signals, would blur this precision. It would smooth out the edges of the night into something imprecise, something the body could not use to time its processes reliably. The precision of the timing is not incidental. It is the point. Melatonin does not merely announce that darkness has come, it encodes the duration of the night, and the duration of the night is information the body uses. A melatonin signal that rose and fell over the correct window of hours tells a different story than one stretched longer or compressed shorter. And so the enzyme that gates the signal must be both responsive and fragile, authorized only by continuous darkness, dissolved the moment that authorization is withdrawn. There is something in the chemistry of the pinealocyte worth sitting with for a moment. Through the bright hours, the cell holds its serotonin reservoir quietly, making no particular use of it. The enzymes downstream of serotonin are present in low amounts or in inactive forms. The cell is not entirely still. It performs its ordinary metabolic housekeeping. It sustains its membranes and its organelles, as all cells do. but the specific chemistry of melatonin synthesis is largely quiet, held in abeyance by the absence of the triggering signal. The cell is a room in which the preparations have already been made, in which the serotonin sits like gathered materials waiting for the work to begin, but in which no work yet proceeds. When the triggering signal finally arrives, when the nerve fibers that approach the gland release their chemical message, and the pinealocyte's membrane receives it, and the cascade inside the cell begins to lift the rate-limiting enzyme's activity upward, the shift is measurable within the hour. The enzyme's activity climbs. The serotonin pool, held all day in reserve, begins to be consumed. The two-step chemistry of melatonin synthesis proceeds. N-acetyl serotonin accumulates briefly, is caught by the second enzyme, is methylated, and exits the cell as melatonin into the surrounding tissue and capillaries. The serotonin pool depletes across the early night, drawn down as the synthesis proceeds. By morning, the concentration of serotonin in the pineal has fallen from its daytime high. This is not a loss. It will be rebuilt the following day as the synthesis pathway, starting from tryptophan, runs again through the long hours of daylight, restocking the reservoir for the next night. The biochemical preparation for each night's melatonin output begins in the morning of the same day. The day and the night are connected through this chemistry more intimately than they appear from the outside. The sunrise that ends melatonin production is also through this replenishment, already beginning to prepare for the melatonin production of the following night. A single melatonin molecule, once released into the blood, survives for roughly 40 to 50 minutes before the liver metabolizes it, converting it through a hydroxylation step to 6-sulfotoxy-melatonin, which exits the body in the urine. This means the presence of melatonin in the bloodstream at any given moment is not the product of synthesis that occurred hours ago. It is the product of synthesis happening right now, continuously within the pineal gland, sustaining the signal moment by moment. The blood concentration at 2 in the morning reflects the current output of the gland at 2 in the morning. The curve of melatonin across the night is not an echo of earlier events. It is a live report of ongoing chemistry. This continuous production also means that the signal can fall quickly when synthesis stops. The half-life of melatonin in the blood is short enough that by the time the first light of morning has reached the retina, traveled its neural relay, and withdrawn the adrenergic signal from the pinealocyte, the rate-limiting enzyme has already been degraded, synthesis has ceased, and the blood concentration of melatonin is already falling. Not gradually, not with the slow tide going out quality of many hormonal signals, but relatively quickly. Within an hour or two of morning light, the melatonin signal has substantially withdrawn, and the pineal gland has returned to its daytime quiet. The melatonin that has already been released into the body continues its metabolic clearance independently of the gland's current state. The liver catches it, modifies it, and prepares it for excretion, processing each molecule on a schedule determined by the molecule's own chemistry, rather than by any further signal from the pineal. So the dissolution of the night is not a single event, but a sequence of parallel processes, the gland quieting, the blood concentration falling, the remaining circulating molecules being caught by the liver one by one, and the urine collecting through the morning hours, the final metabolic record of the night's hormonal output. Researchers can measure that record. The concentration of 6-sulfotoxy-melatonin in a morning urine sample is a trace of the previous night's melatonin production, a chemical impression of how much was made, across what duration, at what intensity. In this way, the urine carries a kind of diary of the night before, written in metabolites, readable by the right instruments. The body keeps its own records of its own chemistry. even though no part of it is trying to do so. The record is simply a consequence of the biochemistry proceeding as it does, the liver's modifications creating a downstream marker as a natural byproduct of clearing the signal. The moment the rate-limiting enzyme's activity peaks, somewhere in the middle of the night, when the blood concentration of melatonin has climbed to its highest point, the pineal gland is producing melatonin at the maximum rate its current conditions permit. In healthy young adults, this peak concentration in the plasma is typically somewhere between 100 and 200 picograms per milliliter. In some individuals, it reaches higher. In others, it falls somewhat short of that range. The variation is real, shaped by age, by genetics, by the degree to which the previous evening's light exposure may have compressed the onset of production. But within any given person, the pattern across nights is relatively consistent. The same approximate shape, the same approximate timing, the same approximate peak, the physiology returning to its pattern the way a river returns to its channel after a moderate flood. All of this begins, as noted at the start, with the quiet crossing of a threshold in the late evening. Dim light melatonin onset, the point at which the sustained rise becomes detectable in the blood or saliva, typically falls roughly 2 hours before a person's habitual sleep time. Not at the moment of falling asleep. Earlier, the chemistry of the night begins before the sleep itself, a preparation rather than a simultaneous event, the hormonal signal advancing ahead of the behavioral one, the way the light of dawn arrives before the sun itself clears the horizon. This timing is not arbitrary. The body does not simply produce melatonin because it is dark and then fall asleep. The melatonin signal is one of the inputs into a system that promotes sleep, and its rise in the early evening is part of a broader shift in the body's state, a lowering of core temperature, a reduction in alerting signals from the circadian clock, a growing balance between the drive toward sleep and the conditions that permit it. Melatonin does not force sleep the way a sedative does. It signals. It informs the body's systems of the time of night, and the body's systems respond to that information according to their own internal logic. The chemistry of sleep onset is orchestrated, not commanded, and melatonin is one voice in that orchestration, early, consistent, heard throughout the body, but not alone. On any given evening, in the hour before the onset becomes measurable, the pineal gland is already beginning to stir. The rate-limiting enzyme's activity is climbing from its daytime low. The serotonin pool is beginning to be drawn upon. The first melatonin molecules are appearing in the blood at concentrations too low yet to cross the threshold of a standard assay, but present nonetheless. A slow advance before the main signal rises. The chemistry is already in motion before any instrument would record it, the way a tide begins moving before the waterline makes it visible. When the threshold is crossed, when the sustained rise becomes unambiguous, when the concentration climbs past the few picograms per milliliter that mark the recognized onset, the night's chemistry is committed. The signal will now rise for several hours, reaching its peak in the small hours of the morning, before declining again ahead of habitual wake time. The decline begins before waking, before the alarm, before the light, before any deliberate return to consciousness. The pineal gland is already tapering its output. The morning's arrival is anticipated in the chemistry several hours in advance. The system's internal clock already beginning to call back the night's signal before the external world provides any confirmation that morning is near. By the time the first light reaches the eyes and the retinal signal travels its relay, the melatonin peak has already passed. The rate-limiting enzyme, still active but at reduced levels, finds its sustaining signal further withdrawn with each passing minute of light exposure. Within minutes, the enzyme's degradation accelerates. Within an hour or two, plasma melatonin has fallen close to its daytime baseline. Within another few hours, it is there, low, stable, the baseline that will persist through the full arc of daylight, and the serotonin pool has begun, quietly, to rebuild itself for the following night. The whole cycle will repeat. It repeats every day. It has repeated, in one form or another, in the ancestors of every vertebrate alive for an evolutionary span of hundreds of millions of years. The chemistry has been modified. The precise regulation differs between species. The magnitude of the nocturnal surge varies. The relative roles of the two enzymes have shifted in different lineages, but the core logic is ancient. Darkness falls, a signal is released, a reservoir is consumed, a molecule appears in the blood, the night is announced in chemistry to every corner of the body, and then, with morning, the announcement is withdrawn. What makes this chemistry remarkable is not its complexity. The synthesis chain is only a few steps long. The enzymes involved are well characterized. The logic of the rate-limiting step is straightforward, a narrow gate controlled by a signal, producing a proportional output. What makes it remarkable is its precision, and the fact that this precision is maintained across a lifetime of nights, reliably, in a gland smaller than a pea, producing concentrations so minute they cannot be felt or seen or sensed by any ordinary means, and yet read without error by every organ and tissue in the body that carries the receptors to receive them. The serotonin that waits in the pineal through the long hours of daylight is not waiting for anything in the sense of a mind's patience. It is simply there, at the concentration the day's biochemistry has brought it to, held by the same cellular machinery that synthesized it. The rate-limiting enzyme that rises and falls each night does not know that it is a gate, does not know what it is gating, does not register the completion of the cycle it makes possible. The melatonin molecule that drifts through the capillary wall and enters the bloodstream carries no awareness of the distance it will travel or the tissues it will reach. None of these events has a purpose in the sense of intention. They are chemistry. They are the body doing what the body does in the dark. And yet, from the accumulated action of that chemistry, the pooling of serotonin, the nocturnal surge of the enzyme, the two-step synthesis, the fragile stability of the rate-limiting protein, the continuous release and rapid clearance, the rise and fall of a picogram scale signal in the blood. From all of this repeated nightly emerges the body's most reliable internal announcement of the night. The molecule that says, darkness is here. The molecule that says, the night has begun and can be measured and will end. The molecule so small it should be invisible, carrying information the whole body depends on. And in the morning, without announcement, without drama, with nothing more than the withdrawal of one molecular signal from one small protein in one small gland at the center of the brain, the rate-limiting enzyme dissolves. Not slowly, in minutes. The night's authorization, extended across all the hours of darkness, simply ceases to be renewed. The enzyme that held it open, that converted serotonin into the molecular step before melatonin, that kept the synthesis flowing through the long interior hours, it fragments. Its pieces are absorbed by the cell's own maintenance systems. The machinery of the night's chemistry is quietly disassembled. The pieces return to the general cellular pool from which new molecules will be built as the day requires. The serotonin pool, depleted now from the night's work, will slowly be replenished. L-tryptophan will arrive again in the blood from the food eaten today. Tryptophan hydroxylase will work again through the morning and the afternoon, producing 5-hydroxytryptophan, which will be decarboxylated to serotonin, which will accumulate again in the pineal's reservoir, ready for the following night. The supply chain for darkness is restocked in daylight, the day working quietly on behalf of the night to come. Outside, in the blood, the last melatonin molecules from the night before are being caught by the liver, hydroxylated, conjugated to sulfate, and carried to the kidneys. They will leave the body in the urine of the morning, a final trace of last night's chemistry finding its way out of the system entirely. By midday, none of them will remain in the circulation. The blood will carry its daytime baseline, a concentration so low it barely differs from zero on the scale of the night's peak. And the pineal gland, quiet now, filled with its slowly rebuilding reservoir of serotonin, will hold the chemistry of the next night's signal in patient, unintended waiting, the same way it has held it every day for every year of a life. One night's preparation, indistinguishable in its chemistry from any other, the body turning its patient, unwitnessed cycle, the same molecule dissolved into the blood, the same molecule fading again with the light, the same dissolution at dawn, arriving precisely on time as it always has and always will, directly above the place where the optic nerves cross, where the visual information from both eyes meets and divides and continues deeper into the brain. There sits a structure so small that it fits comfortably inside a thumbnail, and so densely packed that it contains within its rice grain volume one of the most reliable timekeeping mechanisms in the entire body. This is the suprachiasmatic nucleus. It rests in the anterior hypothalamus, low and central, its position almost architectural in its logic. The light pathways arrive from below, and the clock sits directly above them in perpetual conversation with the world outside the skull. Each half of the nucleus contains roughly 10,000 neurons. Together, the two form a paired structure small enough that the whole of it would disappear behind a pencil point. Yet every one of those 20,000 cells runs, at all times, a molecular program of extraordinary precision. They cycle. They have always cycled. They will cycle through the night ahead, and the one after that, and the one after that, maintaining a rhythm that runs close to 24 hours, whether or not the sun rises on schedule, whether or not any light reaches the eye at all. To stand at this scale, inside the hypothalamus, at the level of a single neuron, is to stand beside a machine that has been running without interruption since before the individual carrying it was born. The ventrolateral core of the nucleus, the region closest to where the optic nerve input arrives, is the part that listens to the outside world. It receives a direct projection from the retina. During the day, that projection carries the signal of ambient light, and the core adjusts. At night, when the signal from the eye quiets, the dorsomedial shell, the outer layer of the nucleus, less directly connected to retinal input, expresses something that runs on its own internal authority. The shell keeps time even when no signal comes from outside. It cycles through its molecular program the way a pendulum swings, not because something is pushing it, but because that is what its nature requires. The molecular machinery inside each SCN neuron is built from a small set of proteins that produce each other and suppress each other in a loop that takes almost exactly a full day to complete. At the center of this loop are two proteins that work as a pair. One is called clock, the other is called BML1. They bind to each other and move together into the nucleus of the neuron, where they act as transcription factors, molecules that unlock certain genes and set them running. The genes they activate are called PAR and KIRI. Each name is an abbreviation, but the important thing is not the name. The important thing is what happens next. The PER and CGRI genes activated by the cluck and BML1 pair, begin producing their own proteins. Those proteins accumulate slowly through the course of the day, building up in the cytoplasm of the cell, like water slowly filling a vessel. They do not act immediately. They take time, many hours, to reach a concentration sufficient to do what they are built to do. And what they are built to do is return to the nucleus and suppress the very proteins that created them. PAR and cry, once they accumulate enough, re-enter the nucleus and inhibit cluck and BML1. They switch off their own production. This is the loop. A pair of proteins activates the genes for a second pair. The second pair accumulates, enters the nucleus, and inhibits the first. With the first pair suppressed, the second pair is no longer produced, and it gradually degrades. As it degrades and its concentration falls, the suppression on the first pair lifts. Cluck and BML1 become active again, and the whole cycle recommences. The cycle takes approximately 24 hours, not because 24 hours is a convenient unit, but because the rates of transcription, translation, protein accumulation, nuclear entry, and degradation in these particular proteins are such that the loop closes in roughly that time. The world outside the skull happens to turn on a 24-hour schedule. The proteins inside the neuron happen to cycle in close to the same period. The match is not coincidental. It is the result of billions of years of selection pressure, so consistent that the molecular clock and the rotating planet have converged on nearly the same rhythm. There is a second stabilizing loop running alongside the first, a quieter mechanism that prevents the primary cycle from drifting or losing its shape over time. Two proteins, one called Uriv-Arba and one called Ro-Arafa, compete for the same regulatory site on the BML1 gene. Reva-Grediva suppresses BML1 transcription. Reva activates it. Through the day, their relative concentrations shift, and their competition at that site adds a layer of stability to the timing, preventing the primary loop from wandering outside its roughly 24-hour range. The whole system is not a single loop, but an interlocked set of loops, each checking the others, each contributing to a rhythm precise enough to set a clock by. And there is one more element of precision. Even if the proteins are produced on schedule, they have to enter the nucleus at the right time for the loop to close correctly. The timing of nuclear entry is controlled by phosphorylation, the addition of phosphate groups to the PER proteins, carried out by enzymes called casein kinase 1 delta and casein kinase 1 epsilon. These enzymes tag PER proteins, and those tags affect the protein's stability. A pure protein that is heavily phosphorylated is marked for degradation. One that carries fewer phosphate groups is more stable and more likely to persist long enough to enter the nucleus when the concentration is right. The kinases are, in this sense, fine-tuning mechanisms. They trim the period of the molecular clock to keep it accurate, the way a watchmaker adjusts the tension of a spring. Inside the SCN neuron, all of these elements operate simultaneously, the primary cloak BML1 loop rising and falling, the stabilizing competition between Reva-Brava and RR, the phosphorylation of PAR proteins governing their longevity. The output of all of this is not a single protein, but a pattern of neural activity, a rhythm of firing and silence that the nucleus generates and broadcasts across the day. During the subjective day, SCN neurons fire at higher rates. During the subjective night, their firing quiets. This electrical rhythm, coordinated across tens of thousands of neurons, becomes the signal that drives the rest of the brain. The question, then, is how this electrical rhythm inside a small nucleus in the hypothalamus becomes the hormonal signal that marks the night for every tissue in the body. The answer requires a pathway, a relay, And that relay is more anatomically complex than its purpose might suggest. The signal does not travel directly from the SCN to the pineal gland. There is no single axon running from the clock to the secretory cells. Instead, the signal descends. It leaves the hypothalamus, and it descends a considerable distance before it rises again. From the SCN, the first connection runs to a region of the hypothalamus called the paraventricular nucleus. The paraventricular nucleus is a hub. It receives input from the SCN and projects widely to other brain regions and to the brainstem. Among its many outputs, it sends a projection downward through the brainstem and into the spinal cord. The signal descends to the upper thoracic spinal cord into a column of cells called the intermediolateral cell column, situated at roughly the first and second thoracic segments. These are preganglionic, sympathetic neurons. They are part of the autonomic nervous system's sympathetic division, the same broad network that orchestrates the body's responses to activity and demand. From the intermediolateral cell column, the signal continues outward along preganglionic sympathetic fibers, traveling through the chest and upward along the carotid arteries into the neck. And there, at the base of the skull on either side, it synapses onto a cluster of neurons called the superior cervical ganglion. This ganglion is a relay station. It sits outside the brain and spinal cord, inside the sympathetic chain, a structure whose position in the neck belies its importance to the brain chemistry it influences. From here, postganglionic sympathetic fibers travel upward along the carotid nerve plexus, accompanying the internal carotid artery as it ascends into the skull. These fibers are fine, unmyelinated threads. They carry no visual information. They carry only one message, the message from the clock. It is night, and the message arrives at the pineal gland. The route is long, SCN to paraventricular nucleus, to intermediolateral cell column, to superior cervical ganglion, to pineal gland. 4 distinct stations, each requiring a synapse, each requiring a neurotransmitter to cross the gap and bind a receptor on the far side. The signal has traveled from the hypothalamus down the length of the upper trunk and back up through the neck into the skull. It has crossed from the central nervous system into the peripheral sympathetic nervous system and returned. It has done all of this to carry one instruction to a gland the size of a pea. When the postganglionic fibers arrive at the pineal, they release a neurotransmitter. That transmitter is norepinephrine. It crosses the synaptic cleft and binds to receptors on the surface of the pinealocytes, the secretory cells of the gland. The binding sets off a cascade inside the cell. The receptor is coupled to a signaling protein, and the coupling increases the intracellular concentration of a messenger molecule called cyclic AMP. Cyclic AMP activates a kinase, and that kinase does several things, among them protecting the rate-limiting enzyme of melatonin synthesis from rapid degradation. During the day, that enzyme, the one that drives the synthesis cascade, is quickly broken down. But under the influence of the norepinephrine signal, in the dark, it is protected, it accumulates, it becomes active, and the synthesis of melatonin begins. What the clock releases then is not melatonin itself. The clock releases a pattern of neural firing that, by the long path described, reaches the pineal gland as a norepinephrine signal. The norepinephrine signal is the authorization. It is the molecular permission to begin the night's chemistry. The pinealocyte receives it, amplifies it through its own internal signaling chain, and responds by sustaining the enzyme whose activity defines the secretory night. This is the architecture of the relay, a single nucleus containing 20,000 cycling neurons, generates a daily rhythm that propagates through a four-station pathway, crosses from brain into peripheral nervous system and back, and delivers its output as a burst of sympathetic neurotransmission to a gland at the center of the skull. The entire chain is as dedicated to its single function as a telegraph line. There is something worth pausing on in that relay, not its complexity, which is easier to appreciate when traced step by step, but its vulnerability. Each synapse along the path is a point where the signal can be interrupted. A disruption at any of the four stations, at the paraventricular nucleus, in the spinal cord, at the superior cervical ganglion, along the postganglionic fibers, would silence the melatonin signal entirely. Certain neck injuries, certain surgeries, certain disease processes affecting the autonomic nervous system can do precisely that. The signal that arrives at the pineal every night by this long and circuitous route is not guaranteed. It is transmitted, synapse by synapse, through a relay that takes nothing for granted. And a healthy individual, in the dark of a night with no artificial light, the relay completes. Norepinephrine arrives at the pineal. The synthesis cascade is authorized. Melatonin enters the blood. And then something occurs that closes the loop in a way that is, on reflection, one of the more quietly extraordinary features of this whole system. Melatonin, once released into the bloodstream, circulates to tissues throughout the body. It reaches the liver, it reaches the gonads, it reaches the gut, it reaches the brain, and among the places it reaches, it returns to the suprachiasmatic nucleus itself. The SCN carries receptors for melatonin. Not many types, two principal ones, called MT1 and MT2. They are distributed across the neurons of the nucleus, and their binding properties and downstream effects are distinct. The MT1 receptor, when bound by melatonin, inhibits the firing of the neuron it sits on. It reduces the production of cyclic AMP inside the SCN cell. It quiets the neuron. This is one mechanism by which melatonin, the output of the clock's timing signal, feeds back to suppress activity in the clock itself during the biological night, a kind of hormonal signal that deepens the quiet it is reporting. The MT2 receptor has a different set of downstream effects. Its activation does not simply suppress the neuron's current activity. It engages signaling cascades that can shift the timing of the molecular clock, the entire cycle of PER and Ca-Ri protein accumulation and nuclear entry, forward or backward, depending on when, during the subjective day, the signal arrives. Melatonin given in the late afternoon can advance the clock, push it earlier, so that evening falls sooner in the brain's reckoning of time. Melatonin given at an unusual hour can shift the phase of the entire rhythm by measurable amounts. Ew. The hormone is not merely a report of what the clock is doing. In returning to the clock, it participates in setting the clock. This is what makes the MT2 receptor's presence in the SCN something more than a simple feedback loop. Feedback, in the ordinary sense, is a mechanism that dampens or amplifies an output. What melatonin does at the MT2 receptor is something more nuanced. It provides the clock with a hormonal reflection of its own prior output, and through that reflection, the clock can be adjusted. The signal goes out from the SCN, travels through 4 neural stations, triggers melatonin synthesis, releases melatonin into the blood, and the blood carries that melatonin back to the very neurons that initiated the sequence. The loop is closed not only biochemically, but functionally. The clock can, through this mechanism, reset itself on the basis of what it has already done. In the context of ordinary life, this is how the circadian system remains aligned with the external day. Light information arrives through the retina to the SCN core, informing the clock of when actual dawn occurred. Melatonin feeds back to the MT2 receptors in the SCN, providing a hormonal confirmation of when darkness fell. Together, these two signals, one entering from outside through the optic projection, one rising from within through the bloodstream, allow the master clock to calibrate itself daily against both ends of the light-dark cycle. In the context of disrupted schedules, travel across time zones, prolonged shift work, irregular light exposure, the same feedback mechanism is why carefully timed melatonin can, in some circumstances, ease the body back into synchrony. The MT2 receptors receive the exogenous signal, interpret it as a report of when darkness fell, and adjust the phase of the molecular clock accordingly. The adjustment is not large. It does not reset the clock in hours. It nudges it, the way a hand pressed briefly against a pendulum nudges the period of its swing, without arresting it. The SCN neurons themselves, beyond their role as recipients of melatonin feedback, are also synchronized to each other. 20,000 neurons, each running its own molecular clock, do not do so in isolation. They are electrically coupled. They communicate through gap junctions and through local neurotransmitter release. Neuropeptides, including one called vasoactive intestinal peptide, play a particularly important role in coordinating the phase of the molecular oscillations across the population of SCN cells. The result of this intercellular coordination is that the SCN does not behave as 20,000 independent clocks running at slightly different rates. It behaves as a single coherent oscillator, its cells mutually entrained to a common phase. The precision of the whole exceeds the precision of any individual cell, in the same way that a chorus holds a note more steadily than a single voice. During the day, with the molecular clocks of the SCN neurons in their active phase, with the nucleus sends signals that suppress melatonin production, not through the long sympathetic relay, but through direct inhibitory projections to the paraventricular nucleus that keep the entire downstream chain from activating. The neural relay is gated, suppressed, held in suspension through all the hours of the subjective day. The pineal is kept quiet. Only when the SCN enters its own subjective night, its firing rate declining, does that inhibitory tone lift, and in that lifting, the pathway opens. The paraventricular nucleus, released from SCN inhibition, allows the signal to descend toward the spinal cord. The relay begins, and the pineal, after a day of silence, receives the permission it has been waiting for. In an environment without clocks, without windows, without any artificial source of light, the body finds a rhythm that is not exactly 24 hours. It is close within a few minutes for most people, but it is not precisely the same as the solar day. If isolated completely from all light information, the free-running period of the human circadian system is on average very slightly longer than 24 hours. Each day, without entraining signals, the body's internal midnight would arrive a few minutes later than the night before. Over weeks of isolation, sleep onset would drift steadily around the clock. Light prevents this drift. The daily projection from the retina to the SCN core allows the clock to reset itself slightly each morning, anchoring its subjective dawn to the arrival of actual light. The melatonin feedback through MT2 receptors contributes an additional nighttime anchor. Together, these two signals, one entering from the environment, one generated internally and fed back, keep the free-running rhythm of roughly 24 plus hours from drifting away from the 24-hour rotation of the planet. The SCN does not merely regulate melatonin. It is the master pacemaker and its outputs reach far beyond the pineal relay. It communicates the time of day to every major organ system in the body through combinations of neural projections, humoral signals, and the regulation of body temperature, cortisol release, and autonomic tone. The melatonin signal it generates is one of several systemic outputs, but it is a particularly far-reaching one, a molecule that enters the blood and circulates to every tissue, carrying the time of night to organs that have no direct connection to the clock in the hypothalamus. The body temperature rhythm, the cortisol rhythm, the patterns of alertness and fatigue across the waking day, all of these are orchestrated in part by the same 20,000 neurons cycling quietly in the anterior hypothalamus. The melatonin signal is the one most legible from the blood, the one most directly tethered to the light-dark cycle, and the one most clearly aligned with the onset of sleep. It is the clock's clearest public statement, the announcement broadcast in a chemical language, readable by every cell that carries the right receptor. There is a quality to this system that becomes apparent only when it is considered as a whole. The molecular clock inside each SCN neuron, the interlocked protein loops cycling over 24 hours, generates a rhythm whose period is set by the rates of biochemical reactions. Those reactions have been tuned over evolutionary time to produce a period close to the length of the Earth's day. The neural relay that converts the clock's output into a pineal signal passes through 4 synapses and descends below the diaphragm before rising back to the brain. The melatonin molecule, released at the end of that relay, returns to the clock that authorized it, binding receptors that can shift the very timing of the molecular loops that started the whole sequence. Each element depends on the others. The molecular clock needs the light signal to stay entrained to the actual day. The neural relay needs the SCN's inhibitory gate to lift before it can activate. The pineal needs the norepinephrine signal to protect its key enzyme from degradation. The melatonin feedback needs the receptor population in the SCN to be present and responsive. The system is not robust despite its complexity. It is robust because of the redundancy built into the coupling between its parts, the mutual entrainment of SCN neurons, the dual receptor system in the SCN for melatonin feedback, the multiple stabilizing loops in the molecular clock. What this means, in the quietest terms, is that the timekeeping system of the body is not a single mechanism. It is a community of mechanisms, each checking the others, each lending its own rhythm to the common signal, and each receiving back from the whole a calibration it could not achieve alone. In the dark hours between midnight and the first light of morning, this community runs at its most undisturbed. The SCN neurons are in the quiet phase of their molecular cycle, their firing rates reduced, their inhibitory projections to the paraventricular nucleus withdrawn. The sympathetic relay is open and active. Norepinephrine reaches the pineal and sustains the synthesis cascade. Melatonin is present in the blood at its peak nocturnal concentrations, circulating to every receptor-bearing tissue in the body and returning quietly to the SCN itself. The SCN neurons that receive melatonin at their MT1 receptors fire a little more slowly. The ones that bear MT2 receptors carry the hormonal imprint of when the night began. The clock that sent the signal to make the melatonin is now receiving the melatonin back, using it to confirm its own position in the day, using it to hold the phase of the molecular loop steady against the following morning. The pendulum swings, the proteins accumulate and dissolve. The signal descends 4 stations and returns through the blood. The clock and the hormone it creates are bound together in a loop that has no true beginning and no true end, only a cycle that repeats, night after night, with the unhurried confidence of something that has been practiced for a very long time. What is left after the relay has completed and the molecule has returned to its source is a pattern of timing that holds the whole body in the same night. Every tissue that receives the melatonin signal, and they are many, is reading the same clock. The liver, the gut, the immune cells, the reproductive organs, the thermoregulatory centers of the brain, all of them receive, in the blood that perfuses them, the same hormonal announcement. It is night. The cycle has reached the dark. What belongs to darkness may now begin. The body coordinates itself through this shared signal in ways that would be impossible if each tissue ran only its own local clock without reference to a common central source. Peripheral clocks exist. Each organ has its own molecular oscillator, but they are set in part by the melatonin tide that arrives each night from the pineal, which is instructed by the sympathetic relay, which is released by the SCN, whose rhythm is anchored to the light that enters through the eye each morning. The chain holds. Each link passes the signal along without losing it, and the signal, when it returns to the SCN from the blood, completes a circle whose circumference spans the hypothalamus, the brainstem, the spinal cord, the neck, the center of the skull, and the blood that connects them all, a circle that the body traces every night, without effort, without awareness, and without fail. The 20,000 neurons continue their cycling. In the ventrolateral core, the molecular loops are completing another revolution, PER proteins, degrading toward the trough of the night, the suppression on cluck, and BML1 beginning to ease. The first faint preparations for the molecular dawn that will arrive in the hours before the eye has seen any light. The clock begins its turn toward morning before morning comes. It has always done this. It knows, in the only sense available to a set of proteins cycling inside a neuron, that morning is approaching, because its own internal rhythm is approaching the part of its cycle that corresponds to dawn. This anticipation, if it can be called that, is one of the more striking features of the molecular clock. The system does not react to morning, it predicts it. By the time actual light arrives at the retina, the SCN is already shifting its output, already beginning to reconstruct the inhibitory tone that will, over the following hour or two, close the pineal relay and end the night's melatonin production. The body begins waking before it is asked to. The melatonin level in the blood will begin its decline before sunrise. The enzyme that drove the synthesis will begin to lose its protection, the norepinephrine signal fading as the sympathetic relay is gated closed by the returning activity of the SCN. The enzyme will degrade quickly, as it always does when its support is withdrawn. The melatonin already in the blood will be cleared, metabolized by the liver, its byproducts excreted. By mid-morning, very little will remain circulating, but the clock has not stopped. It has only completed one arc of its revolution and begun the next. The clock and BML, one proteins freed from the inhibition of the PR and CRY pair, as those proteins degrade through the morning hours, will begin once more to drive the expression of the genes whose products will accumulate through the day and whose accumulation will, many hours from now, produce the conditions for another night, another relay, another hormonal tide. The cycle is continuous. Day does not interrupt it. Day is part of it. The molecular clock does not pause at sunrise and resume at sunset. It rotates without stopping, spending half its cycle in what might be called its day phase and half in what might be called its night phase, and the entire rhythm of the hormonal night, the relay, the synthesis, the secretion, the feedback, is contained inside the night half of that continuous rotation. To know this is to understand the nightly melatonin signal not as an event that occurs in the dark, but as the expression of a cycle that never ceases. The darkness is not what creates the rhythm. The darkness is what aligns the rhythm to the planet. The rhythm itself belongs to the cell and to the proteins inside it, and to the long evolutionary history that tuned those proteins to cycle in the same period as the turning of the world. In the quiet of the hours well past midnight, while the body holds the deepest part of its hormonal night, the SCN neurons are doing what they have always done, cycling, adjusting, holding phase, preparing for the turn that is coming. The melatonin is circulating. The MT1 receptors are receiving it and quieting slightly in response. The MT2 receptors are receiving it and noting, in whatever chemical sense a receptor can note anything, that the night arrived at the time the clock expected. The loop between the clock and the hormone it creates is closed. The clock runs, the melatonin returns, and in that quiet return, the hormone arriving back at the very neurons that sent the signal to make it, something like a confirmation occurs, a chemical recognition that the night is proceeding as it should, that the relay has held, that the timing is intact, and that the long patient cycle of the body's deepest timekeeping continues, unbroken, into the hours still remaining before the light. On the inner surface of the eye, spread across the curved wall of the retina like a faint population at the edge of a map, there are cells that do not see the way the others do. They do not detect edges or movement. They do not distinguish a face from a shadow. Among the 130 million rods and cones distributed across the retinal surface, these particular cells number only in the thousands. Perhaps 1 in every 50 of the retina's ganglion cells carries the chemistry that makes them different. Sitting quietly at the margins of vision, they are built not for the instant, but for the hour, not for the flash, but for the long, slow arc of the day. A single one of these cells, seen up close, is indistinguishable at first from its neighbors. It has a cell body, branching dendrites, an axon that runs toward the brain, but folded into its membrane is a photopigment that responds not to the quick flicker of a pattern, but to the steady press of ambient light across minutes of exposure. The pigment holds the light and integrates it, the way a rain gauge holds rain, not the speed of the drops, but their total weight across the day. When the light is bright and sustained and blue, this cell fires. It fires slowly and keeps firing, and does not stop for as long as the light continues. What it is measuring, with a patience most cells do not have, is whether the world outside is still day. These cells carry the name, intrinsically, photosensitive retinal ganglion cells. The name is a mouthful. What it means is simpler. Here are cells in the eye that respond to light on their own, through their own internal chemistry, without any help from the rods and cones around them. They are their own light detectors, old in the history of vertebrate eyes, and they serve a function that has nothing to do with the sharp-edged world of vision. Their signal travels not toward the visual cortex at the back of the brain, where images are assembled, but inward and downward, toward the hypothalamus, toward a small paired cluster of neurons sitting directly above the crossing of the optic nerves. Their message is not a picture. It is a fact. The sun is still up. Hold the night in reserve. Do not yet release the dark. The photopigment inside these cells is called melanopsin. It belongs to the same ancient family as the visual opsins in the rods and cones, but its absorption spectrum sits at a particular place on the visible spectrum, a region of short wavelengths between roughly 460 and 484 nanometers, corresponding to the blue end of the visible range. This is not a coincidence of chemistry. It is a coincidence of sky. The light that fills the open air on a clear day, scattered from above by the atmosphere, is rich in precisely this wavelength. For hundreds of millions of years, blue heavy, full-spectrum light meant outdoors. Midday, the sun well above the horizon. The melanopsin molecule learned to respond to the color of the sky itself. When a photon in this blue wavelength range strikes a melanopsin molecule, the molecule changes shape. That shape change initiates a cascade, slower and more sustained than the quick photochemistry of the rods and cones. A cascade built through a different signaling pathway, one that does not race to produce a brief neural impulse and then reset, but that maintains a continuous signal as long as the photons keep arriving. The cell does not adapt quickly. It does not tune out the light the way the rods and cones tune out a steady background. It stays sensitive. It keeps counting. And so long as it keeps counting, so long as the signal arriving from these cells tells the hypothalamus that bright blue shifted light is present, the pineal gland, deep at the center of the brain, stays quiet. The chemistry that makes the night signal remains suppressed. The molecule of darkness waits. The suppression runs through a chain of neural stations that the light signal crosses before it reaches the pineal. From the intrinsically photosensitive cells, axons travel to the hypothalamus, where the master circadian pacemaker receives the information that light is present in the world. From there, a descending signal runs down through the brainstem and into the upper reaches of the spinal cord, where it makes a synapse in a region called the intermediolateral cell column. From those neurons, the signal travels further, leaving the spinal cord and entering the body's sympathetic nervous system, traveling upward through the neck inside the superior cervical ganglia, then climbing back up into the skull along the carotid arteries. At the end of this long relay, from eye to hypothalamus, from hypothalamus to spinal cord, from spinal cord out through the neck and back into the brain, the final step is simple. No signal arrives at the pineal gland. The sympathetic nerve fibers that would otherwise release their chemical messenger onto the pinealocytes are quiet, and in their quiet, the pineal remains in its daytime state, producing no melatonin, holding the enzymatic machinery for synthesis inert. This is how light keeps the night at bay. Not by acting directly on the pineal, there are no light-sensing cells inside the gland itself, not in the human brain. Not anymore. The gland is entirely blind. It knows about light only by whether the nerve fibers reaching it are active or silent. When the eyes report brightness, the fibers fall quiet, and the pineal waits. Only when darkness comes, only when the photosensitive cells of the retina cease their reporting, only when the long relay goes still, only then does the silence at the nerve terminal itself become the signal. The absence of suppression is the permission, and the night begins. The light that triggers this suppression does not need to be intense. This is one of the more striking things about the system. Under full sunlight, outdoor illuminance reaches 50,000 lux or more, a lux being a measure of light intensity as the eye perceives it. Indoors on a bright day, a well-lit room might register 300 to 500 lux. These numbers matter less than what lies below them. Studies measuring melatonin output under controlled lighting conditions have found that room-level illumination, light of around 100 to 200 lux, the kind of brightness common in ordinary living spaces after dark, can suppress melatonin production by more than half. Some individuals show measurable suppression at illuminances as low as 10 lux, the faint glow of a dimly lit corridor, a screen viewed from a few feet away in an otherwise dark room. The sensitivity of the system reflects the conditions under which it evolved. For most of human history, And for the many millions of years of vertebrate evolution before our species existed, artificial light after dark was at most a campfire at some distance, producing warm, low intensity, long wavelength light with very little blue content. The system's sensitivity to dim light was not a problem, because the dim light of dusk and twilight was exactly the kind of signal the system needed to detect the transition from day to night. The retinal cells tracking ambient irradiance were finely tuned to the natural gradient of the setting sun, the gradual dimming and cooling of the light as the atmosphere thickened above the horizon. That sensitivity, finely calibrated to the natural world, now operates in an environment almost entirely unlike the one that shaped it. The light of a backlit screen sits in a particular and inconvenient part of the spectrum. Modern displays, phones, tablets, monitors of all kinds, emit light enriched in precisely the short wavelength range that drives melatonin suppression most powerfully. Studies comparing the suppressive effect of different wavelengths have shown that blue light near 460 nanometers suppresses melatonin roughly twice as effectively as green light near 550 nanometers at equivalent photon doses. The biology of the melanopsin system was calibrated to the color of daylight sky, and the screens that have come to fill the evening hours emit, in their cooler, brighter modes, something not unlike a simulation of that sky. A person lying in a dark bedroom, face illuminated by a phone at close range, is delivering to the melanopsin-containing cells of the retina a sustained blue-weighted irradiance that the system has no biological way to distinguish from the continued presence of a summer afternoon. The physiological consequence is not subtle. Evening light exposure of this kind delays the circadian clock. It pushes back the time at which the brain issues the signal that night has arrived. It compresses the duration of nocturnal melatonin secretion. It shifts the dim light melatonin onset, that quiet, measurable threshold in the late evening when the first sustained rise in the hormone becomes detectable, to a later hour. And since sleep onset in humans is closely coupled to the rise of melatonin, the delay propagates forward into the night, into the timing of sleep itself, and carries consequences that extend across the following day. This is not a warning. The body is not fragile, and the system has real resilience. A single late evening of screen exposure is not a catastrophe any more than missing a single night of darkness is. The system recovers, the clock shifts and shifts back. What is worth understanding is the precision of the mechanism that this pattern of light engages. The melanopsin system was not designed with margins for continuous evening exposure to blue-enriched artificial light. It was designed for the clean, reliable transition of sunset, a brief period of dimming and spectral reddening that gave the system unambiguous information about the coming of the dark. The spectral quality of light matters as much as its intensity, and the two interact in ways that are not always intuitive. A bright incandescent bulb, warm, yellowish, long wavelength heavy, suppresses melatonin less effectively at a given lux level than a cooler, bluer fluorescent tube, which in turn suppresses it less effectively than a modern LED display at close range. The melanopsin cells do not respond equally to all photons. They respond most readily to photons in the range of 460 to 484 nanometers, below 400 nanometers in the ultraviolet, and above roughly 520 nanometers, moving toward green and yellow and red. The suppressive effect falls off. Long wavelength firelight, burning orange and red, sits well outside the peak sensitivity of melanopsin. A candle a few feet away in a dark room represents for the melatonin system almost nothing. The warm dim end of the spectrum leaves the night intact, Understanding the action spectrum of melatonin suppression, the precise curve of how different wavelengths of light affect the system, was one of the quietly important scientific achievements of the last few decades. Charles Czeisler and George Brainard and their colleagues, working in the late 1990s and early 2000s, established through carefully controlled experiments that the spectral sensitivity of melatonin suppression matched the absorption spectrum of melanopsin, and that this peaked in humans at approximately 460 to 480 nanometers. This was years before melanopsin had been fully characterized in the human eye. The action spectrum was measured first in the suppression of the hormone, then confirmed in the biology of the cell. The hormone's silence told the researchers what the cell was seeing before the cell itself had been properly described. Those experiments required a particular kind of patience. Measuring melatonin suppression by different wavelengths meant exposing subjects to carefully calibrated, near monochromatic light, single narrow bands of the spectrum at controlled photon doses, then sampling blood or saliva repeatedly through the night to chart the hormone's trajectory. The subjects sat in specially designed rooms. The lights were precisely specified. The conditions were maintained across hours. This kind of work does not generate dramatic moments. It generates curves on graphs, values at specific wavelengths, a shape, the smooth peaked arc of the action spectrum that represents the aggregate response of millions of melanopsin molecules to photons of different energies. Quiet, careful, methodical. The knowledge arrived the way melatonin itself rises, not all at once, but slowly, over time, in the dark. Beyond the daily suppression of melatonin by light, the system encodes something larger. Across the course of a year, as the length of the night changes with the seasons, the duration of the melatonin signal changes with it. In winter, when the nights are long and darkness settles early and lifts late, the period during which melatonin is secreted is longer. In summer, when darkness arrives late and morning comes early, the melatonin episode compresses, a shorter window of hormonal darkness to match the shortened night. The pineal gland is not simply a switch that flips on at dark and off at dawn. It is a clock that measures the length of the dark itself, producing a hormonal waveform whose width encodes how long the night lasted. This seasonal encoding of photoperiod, the ratio of light hours to dark hours across the year, is one of the oldest functions of melatonin in the vertebrate body. In mammals that breed seasonally, the duration of the nocturnal melatonin signal is the primary hormonal cue by which the body knows what time of year it is. Long melatonin duration means long nights, means winter, means it is not yet time for breeding in species that time their reproduction to spring. Short melatonin duration means short nights, means the days are lengthening, means the season is turning. The message is not carried in the height of the melatonin peak, it is carried in the width, in the length of the band of darkness the hormone draws across the night. Humans are not as strictly seasonal in their biology as sheep or hamsters, but the residue of this photoperiodic system persists. Human melatonin profiles do shift across the year, lengthening in winter and shortening in summer, the signal's duration tracking the actual photoperiod of the local sky above the place where the person lives. This signal probably had metabolic meaning across our evolutionary history, shifting appetite, energy use, body composition, in ways that remain an active subject of research. What is clear is that the body has always known what season it is, and the knowledge has always been written in the duration of the nightly melatonin curve, accumulated across weeks of darkness like a slow measurement that the biology reads without effort. In the modern world, that seasonal signal is confounded. Indoor lighting after dark shortens the effective night, regardless of what the sky outside is doing. A person in a brightly lit house in January experiences, at the level of their melanopsin system, a shorter photoperiod than the actual winter sky would produce. The biological winter is truncated. Whether this has meaningful consequences for human health across decades is a question that does not yet have a clean answer. What is known is that the photoperiodic encoding function is real, ancient, and operating, that the body is, even now, attempting to read the season from the light it receives in the evenings, doing its best with information shaped by electric light and glass screens and the long continuity of illuminated indoor hours. There is a measurement that stands at the center of research and clinical practice around the melatonin system. It is called the dim light melatonin onset. The concept is precise. A person sits or lies in very dim light, typically no more than 10 lux, roughly the brightness of a small nightlight at some distance, and saliva or blood samples are collected at half-hour or 15-minute intervals across the late afternoon and early evening. In each sample, the concentration of melatonin is measured. For most of the afternoon, the values are very low. The hormone is absent, or nearly so, from the bloodstream. Then, in the late evening, the concentration begins to rise. Slowly at first, then with gathering steadiness, the curve climbs upward through the baseline. The moment at which melatonin concentration crosses a defined threshold, typically around 3 to 4 picograms per milliliter in saliva, is the dim light melatonin onset. This is the biological beginning of the night. The measurement matters because it is the most reliable indicator the body provides of its internal clock's current phase. The time of the dim light melatonin onset is not the same for everyone. It shifts earlier in people who are constitutionally early risers, later in those whose internal clocks run long and whose natural sleep timing is delayed. It shifts with age, tending earlier in older adults, later in adolescents, whose clocks undergo a genuine biological shift toward later phases in the teenage years. It shifts with jet lag, moving in the direction of travel and returning over days as the clock resynchronizes. It shifts with shift work, with seasonal light changes, with the accumulation of evening light exposure. It is, in a sense, the clock's timestamp, the one measurement that tells a clinician or a researcher what time the body believes it to be, regardless of what the wall clock says. For diagnosing circadian sleep disorders, conditions in which the clock's phase is misaligned with the external day, they are bed put down too early or too late for the body's internal timing. The dim light melatonin onset provides information that no amount of sleep history or questionnaire can fully capture. A person may describe going to bed at midnight and lying awake for hours. What the dim light melatonin onset can show is that their internal clock issues its nighttime signal, not at 10 in the evening as it would in a well-aligned individual, but at 2 or 3 in the morning. The biological night and the social schedule are running hours apart. The measurement locates the mismatch precisely. The measurement also guides treatment. Light therapy for delayed circadian phase is timed to fall in the early morning, when bright light delivered to the melanopsin system can shift the clock earlier. Carefully timed supplemental melatonin, taken in small doses at specific hours in the late afternoon relative to the person's measured onset, can accelerate the clock's phase shift. Neither intervention is as useful without knowing where the clock currently sits. The dim light melatonin onset provides that anchor. It is the fixed point from which interventions are measured, the baseline against which recovery or adjustment is tracked. Jet lag research uses the measurement the same way. sampling melatonin under dim light before a transatlantic flight, and again at intervals afterward to chart the pace of resynchronization as the clock crosses time zones and slowly anchors itself to the new local day. The curve shifts forward or backward depending on the direction of travel and tracks back toward the new local time at a rate of roughly 1 to 1.5 hours per day under good conditions. The dim light melatonin onset makes the invisible visible, a concrete measurable number locating the body's internal midnight in the landscape of the clock. A saliva sample collected in a dimly lit laboratory in the late evening holds very little drama in its appearance. It is a small volume of clear fluid in a labeled tube. What the laboratory instrument finds when it measures the melatonin in that sample, a value of 3 or 4 or perhaps 6 picograms per milliliter, the faintest trace of a molecule present in the blood at concentrations that would vanish in a teaspoon of water, is the body's most precise biological declaration that night has arrived. The measurement is indirect, taken at the margin of a hormone's earliest detectable rise, but it is exact. The body does not announce the beginning of its biological night loudly. It does it quietly, in the way that actual nights begin, not with a sharp edge, but with a slow deepening shift in what is present in the blood, a tide turning by degrees in the dark. The light coming through the window at dusk is doing something specific to the cells at the back of the eye. It is shifting. The sun, as it descends, passes through thicker and thicker atmosphere, and the shorter wavelengths scatter away, and what reaches the eye in the last hour before sunset is redder and warmer and lower in the blue-weighted energy that the melanopsin cells respond to most. The effective irradiance at the wavelengths that matter falls, the melanopsin signal weakens. The suppression that has held the pineal gland quiet through the long hours of the day begins to ease. The nerve pathway from eye to hypothalamus, to spinal cord to neck to brain, quiets in its turn. And in the pineal gland, at the center of the brain, the silence from the nerve terminals becomes at last what it has always been ready to become, the permission to begin. This transition from the long daytime suppression to the nocturnal release of melatonin is one of the most precisely timed events in human physiology. It happens at approximately the same hour each evening, within a margin of a few minutes, for a given individual under consistent light conditions, as reliably as any clock. The timing is not fixed to the clock on the wall. It is fixed to the body's own reading of the light, the accumulated signal from the melanopsin cells across the hours of the day, the slow integration of ambient irradiance, the body counting daylight and subtracting it from what came before, arriving each evening at the same conclusion. The night has come, the dark is here, and in the deepening quiet of a late evening, when the light has faded, when the blue-weighted sky is gone, when the eyes no longer report sustained high irradiance to the hypothalamus, the hormone begins its slow rise, not dramatically, not in a sudden pour, the way water rises in a very still basin, barely moving at the surface, the level climbing degree by degree through the lower reaches of the dark hours, reaching its peak somewhere in the long middle of the night. The concentration is small. A picogram is a trillionth of a gram. 100 picograms dissolved in a milliliter of blood is a quantity that no instrument of the previous century could have reliably detected, and yet it is enough. The entire body, from the heart to the gut to the immune system to the reproductive organs, reads this signal. Every tissue with a receptor for this molecule receives the same message. The dark is here, the body settles, the night has been confirmed. The measurement has been made in laboratories around the world. A person seated in a dim room, a series of small plastic tubes collecting their saliva, a technician recording the time on a log sheet as the sample is labeled and stored on ice. In the data that comes back from the instrument, among the low flat baseline readings of the early afternoon, there is one tube where the value is slightly higher, then the next tube is higher again. The curve has turned, the threshold has been crossed. Somewhere in the brain, without fanfare, without any felt event, the clock has issued its signal, and the chemistry of the night has begun its work. The lab record captures the moment as a number, but the moment itself belongs to a process that runs on a longer schedule than any experiment, an ancient, daily, exquisitely calibrated translation of darkness into chemistry, performed by cells that have been doing this for as long as eyes have existed, and light has defined the shape of the day. The dim light falls, the samples accumulate, the hormone rises, and the body, following a timetable written into the cells of the eye and the neurons of the brain and the molecules of a single photosensitive pigment, recognizes the night for what it is and begins in its own quiet and unhurried way to move toward rest. Deep inside a living cell, in the part of it that never rests, melatonin is present at concentrations the bloodstream never reaches, not in trace amounts, not barely detectable. In the mitochondria, those small, folded, endlessly working structures where food and air are converted into the currency of cellular life. Melatonin accumulates at levels many times higher than those measured in the blood, dissolved into the interior membranes of the most energetically intense space the body contains. The chemistry proceeding there is quiet and invisible. It asks nothing of the sleeping person and requires no witness. It simply continues in the dark as it has continued every night of every life that has ever drawn a breath. The mitochondria generate enormous quantities of free radicals as a consequence of their work. Oxygen, bent to the task of energy production, produces unstable fragments as a byproduct, molecules with unpaired electrons that move through cellular space, seeking to stabilize themselves by taking electrons from whatever they touch. Left unchecked, they damage membranes. They nick strands of genetic material. They alter the proteins that carry out the cell's daily operations. The body has systems for neutralizing them, enzymes and molecules distributed through the cell specifically for this purpose. Melatonin is one of them. It donates electrons to these fragments directly, neutralizing them without itself becoming dangerous. It is one of the few molecules that can do this without generating a new free radical in the process, a quality so useful that the cell keeps it close, concentrated, available at the site of greatest need through every hour of the night. The protective work proceeds without fanfare. A melatonin molecule meets a hydroxyl radical, one of the most reactive fragments the mitochondrion produces, and a transfer of electrons occurs. The radical is neutralized. The melatonin molecule is altered in the process, but the altered form is itself capable of neutralizing another radical, and the metabolite that follows can neutralize one more, a cascade of protection that extracts the maximum possible value from a single original molecule, each step quieter than the last, each step further from the site of origin and deeper into the chemistry of repair. And beyond this direct chemistry, melatonin also reaches into the cell's own repair machinery through pathways that do not require binding to a receptor, that operate instead through direct molecular interaction with gene-regulating proteins. It increases the production of superoxide dismutase, an enzyme that catches a particularly damaging class of free radical before it can propagate. It upregulates glutathione peroxidase, another enzyme of the antioxidant system, amplifying the cell's own defenses at the same time it is adding its own. The night, in this sense, is not only a pause from the demands of waking. Inside every cell, it is a period of protected maintenance, and melatonin is one of the molecules that makes the protection possible. There is something very old in this. Free radicals and the chemistry of oxygen have been a challenge to living cells for nearly as long as oxygen has been present in the atmosphere. The antioxidant systems of modern cells carry the long memory of that challenge, refined over vast stretches of time. Melatonin itself is an ancient molecule, found in organisms far simpler and far older than any vertebrate, serving antioxidant functions in single-celled organisms that preceded by enormous spans of time any structure resembling a pineal gland or a circadian clock. That the same molecule in the most complex organisms ever produced by life still concentrates inside mitochondria every night to do the same quiet chemical work it was doing long before there were brains, before there were nights in any biological sense, carries a kind of weight that numbers cannot fully hold. Far from the interior of any individual cell, the same molecule serves a different kind of role entirely. In mammals that live by the seasons, melatonin does not merely signal the onset of a single night. It encodes the length of the night itself, and through that encoding, it tells the body what time of year it is, with a precision that no other hormonal signal matches. A sheep in the northern hemisphere carries within its blood chemistry a continuous record of the shifting ratio of dark to light, updated each night as the melatonin secretion window expands or contracts in response to the changing photoperiod. In the long nights of winter, the melatonin signal is wide, a broad tide of hormonal darkness that rises early, holds through the long cold hours, and retreats only with the late winter dawn. In summer, when the nights are brief, the same signal is narrow, a compressed band of darkness encoding the short night with equal precision. The reproductive systems of these animals read that signal the way a sailor reads water depth, not interested in the moment, but in the pattern, not in a single measurement, but in the accumulating trend. Structures in the pituitary gland, specifically a region called the pars tuberalis, carry dense concentrations of melatonin receptors, precisely because this is where the seasonal message must be received and translated. When winter melatonin, long sustained, arriving night after night across the short days of the cold season, reaches these receptors in a long day breeding animal, the chain of signals it initiates suppresses the hormones of reproduction. Gonads become quiescent. The chemistry of breeding recedes. The animal's body, guided by light and dark, expressed through melatonin's duration, enters a biological winter independent of temperature, independent of food supply, governed almost entirely by this hormonal calendar. The mechanism running beneath this seasonal translation is intricate and involves several hormonal steps. Melatonin binding in the pars tuberalis influences the production of a hormone there, which reaches the hypothalamus and alters the pulsing release of a signaling molecule that governs the entire reproductive axis, changing the rhythm of pulses from a pattern that sustains reproductive activity to one that does not. The timing of that shift and its reversal as the days lengthen again in spring is written in the duration of each night's melatonin signal, a molecular sentence whose meaning is not in its content but in its length. A deer's antlers respond to this calendar. A hamster's body weight shifts in anticipation of it. Migratory birds, whose melatonin systems encode the season with similar precision, begin the internal preparations for departure weeks, before any environmental cue visible to a human observer would suggest a change is coming. The melatonin signal is upstream of all of it, the earliest entry in a long chain of seasonal instructions, the first word of a message whose full expression takes months to arrive. In this sense, melatonin is not only a signal of the night, it is a signal of where in the year the night occurs, a chemical notation of season accumulated drop by drop across the hours of darkness. The breadth of melatonin's reach extends further still. The gut, that long and densely active organ running through the center of the body, responsible for the daily work of digestion and the continuous negotiation between the body and the vast population of microorganisms living within it, contains melatonin in quantities that dwarf what the pineal gland produces. The cells responsible for this are called enterochromaffin cells, and they are distributed throughout the lining of the intestinal wall, interspersed among the other epithelial cells in their millions. Together, they contain an estimated 400 to 500 times more melatonin than the small gland at the center of the brain, not as a circulating hormone, not as a signal of the dark, as a local chemical produced and absorbed within the tissue itself, acting on neighboring cells in the immediate environment of its production. What drives this intestinal melatonin is not the light-dark cycle, but the feeding cycle, where pineal melatonin rises with darkness and falls with dawn. Gut melatonin responds to meals, to the arrival of food in the digestive tract, to the chemistry of digestion itself. The two systems share a molecule, but operate on entirely different schedules and serve entirely different purposes. Gut melatonin influences the muscular contractions that move food through the intestinal tract. It modulates the secretion of digestive enzymes, and it interacts with the immune tissue densely distributed throughout the gut wall. A large fraction of the entire immune system lives there, and it participates in the ongoing relationship between the body and the gut microbiome, that population of bacteria and other organisms whose influence on digestion, immunity, and the chemistry of the brain is still being slowly mapped. The enterochromaffin cells producing all of this melatonin are acting locally, with paracrine signals absorbed into the tissue within millimeters of where they are released, a parallel economy of the same molecule operating at a scale and for purposes that the circadian system never touches. A person in total darkness produces pineal melatonin on its familiar nightly schedule. A person who has just eaten a meal may produce intestinal melatonin at noon, with the sun high and the blood carrying almost none. The molecule is the same, the source. The trigger, the purpose, and the scale are entirely different. That life has used the same chemical in these two such different contexts, one to mark the hours of the night, one to mark the chemistry of nourishment, suggests something about the economy of biological systems. Useful molecules tend to find more than one home. There is one more reach of melatonin's presence that has drawn careful scientific attention, and it belongs to the long, slow intersection of hormonal biology and the biology of cellular growth. Human breast cancer tissue, held in a laboratory perfusion apparatus that allows blood to be pumped through living tumor tissue under controlled conditions, behaves differently depending on what kind of blood surrounds it. Blood drawn from a healthy woman at 3 in the morning, blood carrying physiological concentrations of melatonin, the kind that course through the sleeping body every winter night, quiets the tumor's chemistry. Its metabolic activity slows. Its uptake of linoleic acid, a fat that tumor cells convert into a compound that drives their growth, diminishes measurably. The tumor, bathed in this blood, grows more slowly. Blood drawn from the same women during the day, carrying almost no melatonin, shows none of this suppression. The tumor's chemistry proceeds at its usual rate. More striking still, blood drawn from women who were exposed to light during the night, light sufficient to suppress melatonin, shows the same absence of suppression as daytime blood, despite having been drawn from women who were technically in a sleep period. The hormone's presence was the variable. The tumor's behavior followed it. These experiments, carried out by David Blask and colleagues over many years, did not demonstrate that melatonin cures cancer, or that it causes it, or that the relationship between nocturnal light exposure and tumor biology is simple in any clinical sense. What they did demonstrate is that physiological melatonin, the concentrations the sleeping body produces naturally every night in healthy darkness, exerts a measurable direct effect on the metabolic activity of living human cancer tissue. The effect is real. It is quantifiable. It operates at the concentrations the bloodstream actually carries, and it is abolished, in the tumor as in the blood, by the same light exposure that suppresses the hormone in the pineal gland. What this means in the full clinical picture of cancer biology is still being worked out, with care and appropriate caution, but the experimental finding itself stands. The blood of a sleeping person carrying the night's melatonin carries with it a chemistry that slows certain tumors. The night, in this sense, is not simply restful for the person sleeping through it. The molecular signal of darkness that the sleeping body carries through those hours is, among many other things, a signal that certain kinds of unchecked cellular growth must slow. The hormone is not the only guardian of that function. It is one of several, but it is real, present, and measurable, and it is abolished by light. Across the full span of a human life, melatonin traces an arc that begins high and diminishes steadily, decade by decade, until the signal that was once robust and steep has become something quieter and lower and less precisely timed. A young child's pineal gland, small and dense and barely calcified, produces melatonin in high concentration. The peak reached in the middle of the night in early childhood can exceed 300 or 400 picograms per milliliter, a concentration so much higher than what an adult carries that the two curves placed side by side on a graph belong to what seem like different biological systems. The child sleeps deeply. The signal that authorizes that sleep is strong. Through adolescence, the peak begins to fall, not catastrophically, not all at once, but steadily as the gland changes and the sympathetic nerve signals that drive it mature. The timing shifts as well, pushed later by a combination of hormonal and neurological changes in the adolescent brain that are not fully understood, but that consistently move the melatonin onset toward a later hour and the morning offset later still, in a pattern that has been carefully documented and that helps explain why the teenager who cannot fall asleep before midnight is not simply resistant, but is operating on a genuinely different hormonal schedule. The signal has moved. The body is following it. Through the middle decades of life, the decline continues more slowly. Pineal calcification progresses. Calcium and phosphate crystals accumulate in the soft tissue of the gland in small angular deposits that appear over decades and do not dissolve. The sympathetic nerve fibers arriving at the gland from the superior cervical ganglion, thin threads running from the neck up through the skull, may lose some of their signaling efficiency with age. The enzyme responsible for driving melatonin production may become less responsive to the adrenergic signal that activates it. Any one of these changes would reduce the output of melatonin. Together, they reduce it significantly, and in an individual in their 7th or 8th decade, The nocturnal peak may have fallen to a fifth or a quarter of what it was in young adulthood, a faint version of what once rose steeply each night, arriving later, peaking lower, and receding earlier. The sleep that accompanies this reduced signal reflects it. The ability to fall asleep at a consistent hour, to remain asleep through the night, to wake feeling rested, all of these tend to erode in parallel with the melatonin decline, though the relationship between the hormone and the quality of sleep is complex and not simply one-to-one. Sleep is the product of many systems, not only this one, but the signal that marks the biological night, that initiates the body's transition towards sleep, that communicates the arrival of darkness to receptors throughout the brain and body, is measurably diminished, and the sleep that follows is in many aging individuals measurably changed. There is something in this that is neither tragic nor reassuring, but simply true, in the way that many biological facts are simply true. The body is a system that changes across time, and the changes are not all losses, and even the losses are part of a long arc that was always going to include them. The melatonin signal that marked 10,000 nights of childhood sleep, that drove 20 years of the young adult's reliable biological night, that told a body's cells across decades when to repair, and when to quiet, and when to hold their chemistry in the slow protection of hormonal darkness. That signal does not vanish. It diminishes. It persists, reduced and altered in the tissue of the gland that first produced it. And the body continues to use what it receives. The pineal gland, small and unmoving at the geometric center of the brain, continues its nightly work into the latest decades of life. The calcium crystals slowly accumulating in its tissue do not end it. The signal that arrives from the sympathetic nerve fibers is smaller than it was, but it is still a signal, and the cells that receive it still respond, producing the melatonin they can produce with the machinery that remains available to them. Less than before, present still. Every night, through the life of the body, this proceeds. The nerve fibers in the neck carry the message that darkness has come. The pinealocytes respond, activating the chemistry of synthesis. The molecule that results, small fat soluble, able to pass through any membrane it encounters, diffuses into the bloodstream and spreads. It reaches the receptors in the brain that deepen sleep. It reaches the liver and the kidney, and the immune tissue distributed through the lymph nodes and the gut. It reaches mitochondria and cells throughout the body and takes up its quiet work of protecting them. It reaches tumor tissue and slows certain things that were moving. It measures the length of the night and records it for any system that needs to know what season this is. It returns, finally, to some of the same neural tissue that issued the signal to make it, arriving at receptors in the brain's own timekeeping structure, and completing a loop of molecular communication that has been cycling in one form or another for far longer than the human brain has existed to generate it. By morning, the signal is gone. The enzyme that drove its production is degraded within minutes of the first light reaching the retina, broken down rapidly, leaving almost nothing behind. Melatonin itself is cleared from the bloodstream with a half-life of less than an hour, metabolized in the liver, and converted into compounds that leave the body in the urine. What was present in abundance at 3 in the morning is, by the time the day is fully established, almost undetectable. The night's chemistry dissolves, the day begins, and the serotonin that will become the next night's melatonin begins accumulating again in the pineal, building slowly through the hours of light toward the concentration that will make the following night's synthesis possible. The long rhythm of it is its own kind of steadiness, not the steadiness of a fixed structure, like a crystal or a stone, but the steadiness of a process that repeats, that has repeated in one form or another through the entire span of vertebrate life on this planet, through the cooling of climates and the drift of continents, and the long successions of organisms that looked at darkness and produced from within themselves a chemistry of response to it. The individual nights are all different. The mechanism is the same. It has been the same for a very long time. There is something restful in that kind of oldness. Not the oldness that is worn and diminished, but the oldness of a process that has been tested so many times across so many different bodies in so many different conditions that its persistence is itself a kind of evidence. The melatonin signal that rises in a sleeping body tonight rose in the body of the first mammal that curled in darkness somewhere in the long ago world, and in the reptile before that, and in the fish before that, tracking the dark in each of them with the same chemistry, encoding the same message. Night has come. Slow down. Repair. Be still. The cells of the immune system that receive melatonin signal tonight are not thinking about this history. They simply respond. The mitochondria, carrying out their antioxidant chemistry, are not aware of the molecule working inside them. The tumor tissue that slows in the presence of melatonin-rich blood does not know what has quieted it. None of the systems involved in this vast, nightly, whole-body choreography have any awareness of participating in it. And yet it proceeds in the sleeping child, in the aging body, in the animal under the winter sky, whose reproductive chemistry is reading the length of tonight's darkness and already beginning to record it in the slow hormonal language of the season. The gut's enterochromaffin cells, responding to the last meal rather than to the dark outside, produce their own version of the molecule for their own local purposes. The antioxidant chemistry inside the mitochondria proceeds through the small hours before dawn. The tumor slows. The seasonal signals accumulate, and in the pineal gland, resting small and still at the center of the brain, the night's work continues at whatever pace the aging gland allows, sending its signal outward into the blood as it has always done for as long as it can. The molecule itself is extraordinarily simple for what it does. Its full name is N-acetyl 5-methoxytryptamine, a compound small enough that dissolved in blood at the concentrations the body produces, it is present in amounts too small to see or weigh or otherwise sense. Picograms per milliliter. A picogram is a trillionth of a gram. The amount of melatonin in the entire human bloodstream at its nightly peak would not be visible to the eye. It would rest on the head of a pin and leave room, and yet this quantity, distributed through every milliliter of blood flowing through the circulatory system, reaching every tissue and organ and cell the blood touches, is sufficient to signal the entire body that night has come, sufficient to initiate the physiology of sleep onset, sufficient to slow tumor metabolism in living cancer tissue, sufficient to mark the season in the reproductive system of a breeding animal, sufficient to protect the mitochondria of every cell against the free radicals their work generates. The relationship between the size of the signal and the scale of its effect is not unusual in biological chemistry. Hormones are generally present in tiny quantities and received by receptors sensitive enough to respond to them, the whole system calibrated to transmit information at concentrations that would seem impossibly small from outside. But melatonin reaches into more of the body than most hormones do, binds in more places, performs more distinct functions, and does so in response to a single environmental variable that has been present on this planet for as long as the planet has been rotating. The difference between light and dark that the body has over long spans of evolutionary time built so much on that one variable. Sleep, seasonal reproduction, antioxidant defense, the coordination of immune function, the slow protection of cellular integrity across decades of nightly exposure is a kind of testimony to how thoroughly the distinction between day and night has shaped life. Every organism that evolved under the alternation of light and dark had to solve the problem of organizing its biology around that alternation. Some used behavioral solutions, some used cellular clocks. Some, like the vertebrates, built an organ at the center of the brain specifically to sense the dark and produce a chemical announcement of it that the rest of the body could receive. The pineal gland, the melatonin molecule, the nightly signal that has been rising in darkness and falling in light since before the ancestors of all mammals were larger than a hand. Tonight, in the quiet of a dark room, in the stillness of a body moving towards sleep, the signal is rising again. The sympathetic nerve fibers have carried their message. The pinealocytes have responded. The synthesis is underway, the chemistry moving through its steps in the interior of cells, too small to imagine and too numerous to count. Melatonin is entering the bloodstream. The concentration is climbing. Somewhere between 2 hours before the deepest sleep and the deepest sleep itself, it will reach its highest point, a peak so small by any measure a waking mind might apply, and so sufficient for everything the body asks of it. The mitochondria are being protected. The gut is doing its own quiet work. The immune cells distributed through the body's tissues are receiving their signal.